In this video we are going to learn glycolysis in detail. If you are new to the channel, then please like, share and subscribe to Easylogy-study made easy for more videos. Glycolysis. Glycolysis, from the Greek glykolysis, meaning sweet, and lysis, meaning splitting, also known as Embden-Meyerhof pathway, is a process in which 1 mole of glucose is partially oxidized into the 2 moles of pyruvate in a series of enzyme-catalyzed reactions. Glycolysis occurs in the cytosol of all cells. It is a unique pathway that occurs in both aerobic as well as anaerobic conditions and does not involve molecular oxygen, i.e., oxygen independent. Step 1. Phosphorylation. Glucose is phosphorylated by ATP to form a glucose 6-phosphate. The negative charge of the phosphate prevents the passage of the glucose 6-phosphate through the plasma membrane, trapping glucose inside the cell. This irreversible reaction is catalyzed by hexokinase. Hexokinase is present in all cells of all organisms. Hexokinase requires divalent metal ions such as Mg2+ or Mn2+ for activity. Hepatocytes and B cells of the pancreas also contain a form of hexokinase called gluconase, hexokinase D. Hexokinase and gluconase are isozymes. Gluconase is present in liver and B cells of the pancreas and has a high K and Vmax as compared to hexokinase. ncert is a trivial term for enzymes that are ATP-dependent phosphotransferases. Step 2. Isomerization. A reversible rearrangement of the chemical structure. Isomerization moves the carbonyl oxygen from carbon 1 to carbon 2, forming a ketose from an aldose sugar. Thus, the isomerization of glucose 6-phosphate to fructose 6-phosphate is a conversion of an aldose into a ketose. This step is catalyzed by the enzyme phosphoglucoisomerase. Step 3. Phosphorylation. Fructose 6-phosphate is phosphorylated by ATP to fructose 1,6-bisphosphate. The prefix bis in bisphosphate means that 2 separate monophosphate groups are present, whereas the prefix D means that 2 phosphate groups are present and are connected by an anhydride bond. This irreversible reaction is catalyzed by an allosteric enzyme phosphofructokinase 1, PFK1. Step 4. Cleavage. The fructose 1,6-bisphosphate is cleaved to produce 2 3-carbon molecules glyceraldehyde 3-phosphate, G3P, and dihydroxyacetone phosphate, DHAP. This reaction is catalyzed by enzyme aldolase. Step 5. Isomerization. Only one of the 2 triose phosphates formed by aldolase glyceraldehyde 3-phosphate can be directly degraded in the subsequent reaction step of glycolysis. However, the other product, dihydroxyacetone phosphate, is rapidly and reversibly converted into glyceraldehyde 3-phosphate by the enzyme triose phosphate isomerase. Step 6. The 2 molecules of glyceraldehyde 3-phosphate are oxidized. Enzyme glyceraldehyde 3-phosphate dehydrogenase catalyzes the conversion of glyceraldehyde 3-phosphate into 1,3-bisphosphoglycerate, 1,3-BPG. The reaction occurs in 2 steps. First, the oxidation of the aldehyde to a carboxylic acid by NAD*, and second, the phosphorylation of carboxylic acid by inorganic phosphate, not by ATP. Iodoacetate is a potent inhibitor of glyceraldehyde 3-phosphate dehydrogenase because it forms a covalent derivative of the essential Sh group of the enzyme active site, rendering it inactive. Step 7. In this step, high-energy phosphate group is transferred from 1,3-bisphosphoglycerate to ADP. This step is catalyzed by enzyme phosphoglycerate kinase. The formation of ATP is referred to as substrate-level phosphorylation. In case of substrate-level phosphorylation, synthesis of ATP or GTP occurs by direct transfer from a high-energy substrate to ADP or GDP. Formation of ATP from ADP by direct transfer of phosphoryl group from a high-energy compound is termed as SUT-level phosphorylation. Erythrocytes poses a unique glycolytic bypass for the production of 2,3-bisphospho, 2,3-BPG, the Rappaport-Lubring shunt. This shunt bypasses the phosphoglycerate kinase step and accounts the synthesis and regulation of 2,3-BPG levels that decrease hemoglobin's affinity for oxygen. Step 8. The remaining phosphate ester linkage in 3-phosphoglycerate, which has a relatively low free enter hydrolysis, is moved from carbon 3 to carbon to form 2-phosphoglycerate. Step 9. The removal of water from 2-phosphoglycerate creates a high-energy enol phosphate linkage. The enzyme catalyzing this step, enolase, is inhibited by fluoride. Step 10. The transfer of the high-energy phosphate group that was generated in Step 9 to ADP forms at substrate-level phosphorylation. This last step in glycolysis is the irreversible transfer of the phosphoryl group phosphenol pyruvate to ADP is catalyzed by pyruvate kinase. Pyruvate kinase requires K** and either MgO or Mn2+.